Emerging imaging technologies for non-invasive optical biopsy of human skin. Several high-resolution optical imaging technologies that can be used for non-invasive subsurface skin imaging have recently emerged for real-time detection, assessment, and therapeutic monitoring of skin diseases. These techniques include optical coherence tomography, OCT, reflectance confocal microscopy, RCM, and multiphoton microscopy, MPM. Each technique offers unique information about skin based on imaging depth, resolution, and image capture time. The main advantages of OCT, RCM, and MPM for skin imaging are their ability to provide non-invasive, label-free optical biopsies beneath the skin surface. OCT provides information on overall skin structure, blood vessels, and hair follicles. This technique generates 3D images of tissue microstructures by measuring backscattered and back-reflected light in a way that is similar to an ultrasound, but using light rather than sound. OCT image capturing is rapid and allows deep imaging penetration over a large scanning area. OCT has been used in skin research and clinically implemented to assess non-melanoma skin cancer, evaluate alopecia, and assess vascular skin conditions. Compared to RCM and MPM, OCT has limited spatial resolution imaging and does not offer molecular contrast. RCM provides information on cellular structures of the epidermis and dermis, fibrillar structures of the papillary dermis, and microcirculation. This technique provides subcellular resolution images based on the differences in the refractive indices between different tissue components. It uses near-infrared light from a diode laser as the excitation source. The focused laser light is raster scanned on the skin surface, and the backscattered light is collected to provide grayscale images on reconstruction. RCM allows subcellular lateral spatial resolution over a large scanning area. Limitations of RCM include limited penetration depth and field of view. There is also an increase in imaging time compared to OCT, tens of seconds to a few minutes for RCM compared to a few seconds for OCT. RCM does not offer molecular contrast, but can provide a macroscopic view of lesion morphology with subcellular resolution, including for visualization of melanoma and non-melanoma skin cancers. MPM provides information on structures of the epidermal and dermal cells, collagen and elastin of the papillary dermis, and metabolic changes. Like RCM, MPM is also a laser scanning microscopy technique, but with a different contrast mechanism. MPM images can be multicolor and can distinguish cellular features from extracellular matrix and resemble histologic sections used in diagnosis by dermatopathologists. MPM features submicron lateral spatial resolution and allows molecular contrast, but within a limited field of view. Total imaging time can take 10 to 20 minutes. So far, MPM has only been utilized for dermatological research resolving cellular features in BCC and nevus nests, dendritic and immune cells, melanocytes, and collagen and elastin fibers. Because each of these techniques possesses unique benefits, many clinical applications may require incorporating more than one of these imaging tools. For example, MPM and RCM can be combined for imaging morphological cellular changes in lesions such as melanocytic nevi and melanoma. While both technologies have sufficient spatial resolution to capture upward migration of melanocytes, RCM can provide the assessment over a large area, while MPM would add information related to metabolic changes. For assessing the depth of penetration and extent of basal cell carcinoma, OCT combined with either RCM or MPM would be the best choice because OCT captures the overall morphology, whereas RCM and MPM can visualize the cellular structures inside BCC nests. While these are all promising technologies, there are ongoing efforts to improve upon and overcome their limitations. Collaboration among engineers, scientists, and clinicians is essential for identifying clinical needs and the right imaging approaches to address them.